today's masterclass. My name is Arne and I will be the host for this Pain Emotion masterclass session. We are very happy to have you all here with us at the VUB and a warm welcome to everybody following online as well. It is my great pleasure to introduce today's excellent speaker, Professor Andrea Polli. Thank you so much for being here today and for generously sharing your time and expertise with us. Professor Andrea Polli is a research professor at the VUB and a senior postdoc research fellow at the FWO and affiliated at the VUB and KU Leuven in Belgium. Within the Pain Emotion Research Group, Andrea leads the research line on pain and biology, centered on understanding the complex biological mechanisms underlying chronic pain and related disorders, but with a special or particular focus on the role of epigenetics and DNA methylation in shaping pain responses. Andrea has built a strong track record and he is considered one of the few emerging international experts in the field of pain epigenetics. Today's masterclass topic, the integrative biology of chronic pain from mechanism to personalized care. Professor Polli will speak for about 15 minutes and afterwards we will then have time for a discussion and questions. Please save your questions for the Q&A afterwards and raise your hand when the time comes, please. Professor Polli, the floor is yours.
Thanks, Arne. 15 minutes, right? Not 15. 15. The what?
The mic is very...
Is it for you or for people online? That should be okay, no? Should be fine.
Can we check it? Who is the contact? Let's say it's better now.
Okay.
Sure.
All right, I start anyway. So thank you, Arne, for the introduction and to the rest of the organizing team of these masterclasses for the requests and for the topic that they wanted me to talk about. When I had to prepare this lecture, I started by trying to imagine the future of chronic pain management and what the future of acute and chronic pain management would be in, say, 30 to 50 years. And I think that integrating the biological knowledge that we have now with the bioinformatics and the machine learning models that we have available at the moment, we might really expect in 30 to 50 years to have a real personalized care for people with chronic pain, where with a minimally invasive tests similar to blood pressure monitoring, we can really have a specific personalized approach to assessment and treatment. But to do so, we need to understand pain better, and I think, and I posit, that using the omics data that we have available now, and they're more and more available in the next, will be more and more available in the next couple of years, and integrating them with a multimodal clinical biopsychosocial assessment will do the trick. So I started to think what I should have included in the lecture, and I think the aim of the masterclass is pretty ambitious, because what I'd like to talk about, or at least mention, is the main biological factors that should be integrated to reach a better understanding of pain, particularly persistent pain. We will touch upon genetics, neuroimmune interactions. We will try to integrate environmental and lifestyle factors with biological functions, and then bring everything together. So the first part would be on genetics. So let's start from here. So chronic pain is heritable. Okay, everything I would say started from people observing that children of people with chronic pain have higher chances to develop chronic pain themselves. That doesn't say much about whether this association is due to genetics or due to the environment they are grown in. But afterwards, people started to look a bit more carefully to this link, and they employed what is probably the gold standard for heritability studies, which is twin studies. So what they did was basically comparing identical twin with non-identical twin, and they checked, in this case, it can be done, of course, for any disease or any condition. In this case, is for chronic pain. And the idea is when you compare identical twins to non-identical twins, you have a model where the environment is rather fixed in both identical and non-identical twins. The only thing that changes is the genetics, which is exactly the same in identical twins. It's not as much in non-identical twins. Non-identical twins, of course, are like brother and sister born at the same moment. So if you go on and compare the prevalence of a disease like chronic pain, you can have an idea whether chronic pain is more genetics or more linked to the environment, for instance. And the answer is here. They checked several different chronic pain conditions, and for some of them, like chronic widespread pain, migraine, fibromyalgia, etc., genetic can explain up to 50% of pain in this population, which is quite a good percentage. Then, of course, they try to explore if there is a gene for pain, right? Because that's the logical next question. And of course, they didn't find one specific gene. There is some genes that, if they are mutated, if they change, they give a clear, dramatic change in the clinical phenotype. For instance, think about congenital insensitivity to pain, where the variation of one gene that encodes for an ion channel in sensory neurons will give rise to a dramatic change in clinical phenotype. These people cannot feel pain. But with these rare exceptions, these rare genetic diseases, most, as it happens in nature and in health and disease, what happens is that there are thousands of different variants that can be associated with a condition, and in this case, chronic pain, but these variants normally have low effect size. Okay, so even if you have one of those variants or multiple of those variants, your phenotype, your clinical presentation doesn't change as much. And this is why we call chronic pain as a complex trait. It's not a genetic disease, it's something that where multiple factors, genetics and non-genetic factors co-occur to give the clinical presentation that we know. Another model for studying genetics in chronic pain is the use of genome-wide association studies, which is basically a sequencing of the entire genome, looking for single nucleotide polymorphisms, or SNPs, and see whether these SNPs, these polymorphisms, this little mutation, variation of the genome are associated with chronic pain. And the field is in its infancy. It was the first genome-wide association study on chronic low back pain is dated 2018, so only really a few years. But the field is also expanding very rapidly. So there's a recent systematic review of different genome-wide association studies has been published, including all populations, neuropathic pain, chronic low back pain, CRPS, fibromyalgia, pain after cancer, and so on. And they had this systematic review, and they found a number of genes that have been replicated in multiple cohorts. And they highlighted 6 of them that have been replicated in different studies, different populations, and they also are replicated in the known databases, available databases on chronic pain. And the list of genes are these ones that you can see here. So one conclusion, one speculation that we can already start doing is that they're all related to the immune system, the nervous system, stress responses, descending modulatory mechanisms, and so on. Of course, if you want, you can go read the systematic review. You will also find results per different clinical presentation, but this is a bit the idea, right? Genetics, but also biology in general can help understanding more about one condition, but it can also help understanding what mechanisms are implicated across different conditions. Because of course then the diagnosis is normally clinical, so it's normal that some mechanisms can play a role across different conditions. Very recently, this year, a very large genome-wide association studies has been published on fibromyalgia. I think this is the largest GWAS present, over 2,500,000 people, 55,000, almost 55,000 cases of people with fibromyalgia. And what they found was a number of genes. So what you see here, these graphs, it's the number from 1 to 22, and X is the chromosomes. Every line, every dots that you see is a different gene, and this is the threshold for statistically significant results. So everything that you see above are the number of genes and the name of the genes that survived multiple testing corrections. So these are the genes that seems to be associated strongly with fibromyalgia, and they found a number of different interesting findings. Okay, so the genetic correlation with other chronic and psychiatric disorders, as we expected, over 0.7 of correlation coefficient. They didn't find any sex difference in genetics architecture between men and women, even though almost 90% of the people included were women. But they also, this is what I think it's really interesting from this paper, which is still a preprint, so didn't undergo peer review yet, but they also used external reference data sets to check whether these genes that you see here are associated with mechanisms and processes in specific tissues. And they found that they are mainly, if not exclusively, associated with genes in the nervous system. So the conclusion that this study is proposing is that the fibromyalgia can be defined as a central nervous system disorder, at least from a genetic point of view, which is important. Another study that is really recent, not on fibromyalgia, but on multi-site chronic pain, comes from the UK Biobank, so this large cohort of people that can be studied. They collected large information about clinical data, but they also have biological data, they have the DNA, they have genetics, and so on. And they also performed a GWAS, so a genetic association study, and they found first that about 15,000 or maybe more, 16,000 different genes are associated with multi-site pain. That's many of them, like 11,000, are shared between men and women, but there are about 5,000 that are specifically associated with women, and about 1,000 specifically associated with men. Okay, men unique variations. And they confirm what they found in the previous study on fibromyalgia, because if you see, they also used an external data set where transcriptomics data is available, and they try to see whether the genes that they found were associated with specific mechanisms in specific tissue. And they found, again, that it's mostly central nervous system that is associated genetically with chronic multi-site pain. So this is to wrap up a little bit the first chapter, the genetic part. Try to summarize a few studies, but big studies recent studies that can say something about genetics of chronic pain. And from twin studies, we know that genetics can explain up to 50% of chronic pain variants, especially migraine and headache, chronic widespread pain, and so on. We know that, of course, the field is in its infancy, very recent publications. We know that persistent pain is a complex trait, so many genes might be involved, and you're looking for me? Like this? Yeah. Okay. I've had enough sushi. I don't want to eat this. But okay. And where was I? Vulnerability to chronic pain is likely mediated by nervous system related variation, at least from a genetic point of view, as we said. But we need to be aware that if it's true that there are a number of genes that are replicated and we found significant across different populations and conditions, most others are non-replicated. So there are, of course, methodological challenges when these population studies are employed, mainly related to how you define and who you include in your study, the sample size, because for GWAS studies, you need hundreds of thousands of different participants, of course. But let's go on chapter 2, because we saw that as we knew, of course, I don't think it's a surprise for any of you that the nervous system is highly implicated, but I think some part needs to discuss at least a bit neuroimmune interactions. Okay, so this is just to say that nervous system changes in chronic pain are diverse. They can happen in the peripheral system. They can happen in the spinal cord, especially in the dorsal horn of the spinal cord, and they can happen in higher centers. And if you read this very limited number of mechanisms that I've listed, and you're not familiar with one or more of them, I would recommend to have a look, to know what neurogenic inflammation is, to what hyperalgesic priming is, what neuropathic mechanisms are, but of course also long-term potentiation, central sensitization, descending modulation, and so on. From here, of course, I cannot discuss all of them, and in fact, I am focusing now on neuroimmune interaction, because as you see here, glial cell activity are present and have been found alterated both in the spinal cord and in higher centers. And I think it's interesting and important because until 25 maybe years ago, glial cells were mainly thought as supportive cells. Okay, so cells that were in the nervous system, but they were to support neurons, and neurons were the ones that did all the work, right? But we now know that the nervous system and the brain in particular maybe is a neuroimmune organ, right? And glial cells, mainly microglia that you see here and astrocytes, are really important for any nervous system mechanism. So when we say that the nervous system is implicated, it's not just neurons, it's neuron, glial cells, and of course astrocytes are linked and cover the endothelial cells as well and the blood flow, so the endothelial cells are there as well. We now know that it's not really just about supporting neurons, but as you see here, microglia and astrocytes, they can express the same receptors on the cell that you find on neurons, so they can be activated by the same neurotransmitters, growth factors, inflammatory markers, as they are the same that activates neurons, for instance, and they can express inflammatory markers. Basically, microglia are the and astrocytes as well, are the residents, the immune cells that are resident in the nervous system. So they really work as immune cells in the nervous system. And of course, they express receptors on the cell wall for catecholamines, for instance. So stress can induce changes in microglia and astrocytes. Inside the cell, like in neurons, you might have in the cytoplasm the receptor for glucocorticoids. So cortisol can go through the cell membrane and reach its own receptor in the cytoplasm and then act as a transcriptor factor in the nuclei. Okay, really complex interactions. I might want to say that it's maybe the population that is more related to persistence of nociceptive mechanisms in the long term, more than neurons. In animal studies, this has been known for a while, but in humans, it's of course very difficult to assess, and I think, to the best of my knowledge, the Banati study in 2001 is the first study who did it, who looked at microglial activations in the thalamus after nerve injury. As you can see here, is only 7 people, and they used a tracer that is about 80 times less powerful and specific than what they can use today. But what they did was they found some patients with a nerve injury that had a nerve injury in the past, sometimes some of them a couple of months ago, some of them up to over 1 or 2 years after nerve injury. And what they found, they scanned their brain and they looked for microglia activation in the thalamus. And as you can see, in the ipsilateral thalamus, microglia are expressed, but in the contralateral, which is where you expect them to be overexpressed if they're activated, it's always increased. With the exception of this one, of this patient here, patient number 7, but this also was the only one with a bilateral injury. So it was a spinal cord injury. And this very work started basically an entire field where people started to focus on a neuro-immune interaction in the nervous system because of their importance in maintaining nociceptive activity. People started to even ask whether chronic pain can be a gliopathy, for instance, because most chronic pain conditions show evidence of glial activations. As we said, when they're activated, they can release inflammatory markers, they can release growth factors, they can sensitize neurons, they can remain activated for a long time. Think about them as immune cells, and they learn, and when they're activated, they can remain activated for long. They change their structure, microglia start to have projections, and they are attached to neurons, and they can remain activated for a very long time. And they found glial activations in several parts of the brain: prefrontal cortex, amygdala, hippocampus, thalamus, as we said, etc. And as I said earlier, as they have receptors for catecholamines on the cell wall and they have receptors for glucocorticoids inside the cell, stress can activate them as well. It's not just peripheral mechanisms and peripheral nociception that activate them. It can also be stress, it can be sleep deprivation, and so on. Of course, the hypothesis of glial mediated nerve inflammation has some criticism. The tracer that they use to assess the translocator protein, for instance, which is probably one of the main proteins, the one that is most studied to detect microglia activation, is not too specific for microglia only. Other cells can express it, some neurons, some peripheral immune cells that can infiltrate the nervous system. Astrocytes can express the translocator protein, so it's difficult to be very specific about microglia activation using a scan, that's for sure. And also when they find activation of glial cells, they cannot really tell what subtype of microglia they are looking at. So it might be pro-inflammatory, but it can also be anti-inflammatory. So the field is really trying to understand how to differentiate the function of microglia activation that you're seeing. And last but not least, I would say that neuroinflammation is not pain specific. Parkinson's disease, dementia, schizophrenia, brain trauma, they can also show signs of neuroinflammation, okay? Just to have a balanced overview. But for sure, it's a new player in the field. It's not just about neurons. It's immune cells that can be activated, that can stay activated for long, they can sensitize neurons, and they are definitely maybe the key player that needs to be understood to understand pain better. This was it for central neuroimmune interaction. I wanted to also discuss a bit about the peripheral immune system, because recent studies, interesting studies, have been published, and I normally like to report this one from Luda Dyachenko group at McGill. What they did was they took about 100 people with acute low back pain, and they assessed with transcriptomic analysis, so gene expression, okay, RNA, at the time of the acute injury and after 3 months, okay, and then they compared. They divided the group in two. Half of them recovered, okay, so people who recover after 3 months, and half of them were persistent pain patients after 3 months. And what they found was that in people who recovered, there is a much bigger change in transcriptomics than people who didn't recover. And they could explain it by looking at the RNA expression, that it was mostly an immune reaction led by neutrophils that increased the inflammatory response, and this neutrophil-mediated elevation of inflammatory response, it was then predicted whether somebody recovered or not. And they also, you can also see here, there is neutrophils that increase and then they decrease after 3 months, but there's a big change. So this decrease here means that there was also an increase at time 0, while T cells, CD8 positive, so cytotoxic T cells, did the opposite and increased at T1. And they did more than that, because after enrolling those 100 people, they also went and have animal studies, and they went looking into the UK Biobank. And what they found was that in animal studies, blocking neutrophils would make the animals perceive pain for a follow-up, let's say. And the UK Biobank, what they could find is that the use of non-steroid anti-inflammatory drugs, so anti-inflammatories, increase the risk of reporting pain 2 to 6 years later. So this speaks to the importance that acute inflammation has for the recovery of pain in longer term, right? So it's tricky to stop inflammation with anti-inflammatory in the acute phase. It can be counterproductive. This also goes and adds to other studies in animals and humans, showing that, for instance, giving anti-inflammatories early after acute injury, they can reduce the recovery of bones, of connective tissues, and so on. There are a number of studies that I think are quite interesting, and one of them is this one, I think. So you normally don't see many animal model of fibromyalgia, because we don't know how to induce and we don't know if it's true, if it's correct, the animal model that we use. But these two studies did something very clever, I think. They took blood from patients with fibromyalgia, and in this case here they extracted and purified immunoglobulins, okay, from fibromyalgia patients, and they injected it into mice. And only mice who received the injection of immunoglobulin G from patients started to show pain behavior, allodynia, and they have an increase they started to move less. Okay, it's really something that looks like a patient with chronic pain that moves less, it's hypersensitive, and so on. Similar study, another group, instead of immunoglobulins, they injected neutrophils this time from patients with fibromyalgia, and they also found the same, right? So when you induce neutrophils from fibromyalgia patients, but not from healthy controls, in mice, you see that mice start to behave, to show pain behavior, but also hypersensitivity to external stimuli, allodynia, hyperalgesia, and so on. And here I'm taking a brief detour from the main flow of the presentation, because I was talking about the peripheral immune system, even though, of course, we know that the gut and gut microbiome has connection with the peripheral immune system, among other things. But they did the same with the same idea that we just saw with immunoglobulins and neutrophils was also done with the microbiome on animals, and they found a bit the same results, but I think what they did was even a step further. So they tried to do the same idea in humans as well. So what they did was they took 3 healthy female donors, they collected the stool, they made a pill out of it, so they collected microbiome from healthy donors, and they gave it to 14 people with fibromyalgia, okay? The definition of a shitty study, I would say, because they took the stool and they gave it to people. But it's a shitty study, but it's a good study, so not all shitty studies are bad necessarily. It's about shit, but it's not too bad. And what they found was that in just a week, not all of them, not all patients, but many of them, in just a week, they reduced pain intensity, they reduced the impact, anxiety, depression, sleep disturbances, and so on. Okay? This is not really focusing on the neuroimmune interaction at this stage, even though the authors also also did it in animals, and they found that elevated cytotoxic T cells and interleukin-17 in mice getting the stool from fibromyalgia patients only. But still, it's the same methods that the other ones used. Okay, and I think it's an interesting and clever approach and method to study the importance of the peripheral immune cells, immunoglobulins, neutrophils, and in this case, the gut microbiome. Another important aspect I would think about that is a hot topic in chronic pain and chronic pain biology is sex differences in pain and pain hypersensitivity. This is normally, everything started thanks to the work of Jeff Mogil from McGill again. And when they started to compare men and women or male and female animals, they started to find a number of important differences. Differences in peptides, different in transcriptomics, different in hormones, and different also in immune cells. So for instance, orofacial sensitivity that is induced by a peptide that is called CGRP induce sensitivity. There's also evidence for other parts of the body, but also in female mice, okay? And when they reanalyze clinical data on the effectiveness of CGRP antagonists for migraine, they saw that they worked only in women. Okay, this is important. We need to take into account sex differences, and they propose to consider sex as a biological variable for this matter. They also found that some hormones, like prolactin receptors, were found only in female nociceptors. And there is an interaction between prolactin and this peptide that we just talked about, CGRP. Okay? And also, what they found, always from the same group, it's the work from Sorge. It's a couple of different papers that they were published about 10 years ago. It's that mechanical hypersensitivity in male mice is associated with microglia activations, but in female, sensitivity is associated with cytotoxic T cells, CD8 positive T cells, that release leptin and go on sensitizing neurons. So this is a bit to summarize the sex differences in the immune interaction. As you can see here in female mice, its hypersensitivity and pain sensitivity is mediated by cytotoxic T cells that release leptin, and they go on through the leptin receptor to sensitize neurons. In male, it's microglia that are activated through a purinergic receptor, releases BDNF, which is a growth factor, and also go on through its own receptor to sensitize neurons. There are other mechanisms that are probably true in both male and female mice, like a subtype of microglia, okay, CD11 positive, which is something that they found this year. Okay, the study from FUN 2025, I think it's published in Neuron, discovered this subset of microglia cells, CD11 positive, that are implicated in then neuroimmune interaction in pain. And this was a bit the part, as you can see, it's really fast, and just mentioning a number of mechanisms that are hot topic in chronic pain biology at the moment. And to summarize what we've discussed, it's that the brain is a neuroimmune organ, very important. Neuroinflammation is present in most chronic pain conditions and should be studied. It's probably an important group of mechanisms that should be really understood to get new treatments, new ideas on how to treat persistent pain. Peripheral immune system influence pain through different mechanisms. We've seen the immunoglobulins that are released by B cells. We saw the neutrophils and the importance they have, especially in the acute phase, to induce an appropriate inflammatory response. And that sex is a biological variable. There are many variables, many biological. I only mentioned the 2, 3 that are the main ones, but there are tens of different mechanisms that have been shown to be different between men and women, or female mice, female animal, male animal. Okay, and there is evidence, accumulating evidence, preliminary evidence from studies in humans on gender, that this is in line what we see. There are big biological differences that should be taken into account when we approach and we try to treat pain appropriately. There's no time to go into them, of course. The third part that I wanted to discuss and mention at least is about environmental and lifestyle and how they are integrated, of course, always from a biological point of view. And I would like to start from here, from the importance of childhood adversities. So there are a number of studies now. This is a systematic review from 2 years ago. There are maybe 10 or 20 other studies that have been published in the last 2 years that show, and they are in line with each other, and they show that childhood adversity, so early life stress, can predict a number of things in adulthood, psychiatric disorders, psychological aspects, and chronic pain as well. And of course, the idea is that by changing the stress system and how we biologically react to a stressful situation early in life, this is something that can have an influence in the adulthood. And there is plenty of research going on that seems to support this idea, okay? I wanted to mention it because it also gives an idea of the complexity. Now we've seen different mechanisms, genetics, a number of nervous system related mechanisms that we couldn't really go into, neuroimmune mechanisms, adverse childhood and early stress. But the thing is, they probably interact with one another, right? And if you see at this study, this is also a study done by using the UK Biobank, so 36,000 people. What they did was they assessed different layers of understanding at once. So on the y-axis, you can see the number of adverse childhood experiences, so from 0 to 4. On here, in the x-axis, you can see the hippocampal volume, and we've seen in the list of nervous system related mechanisms, there was one about the gray matter volume in the brain, and for sure one region that has been associated with the persistent pain is a reduction in hippocampal volume. And the three different lines are whether those people had trauma, a single trauma, or multiple trauma early in life. Sorry, here it's not the multiple trauma, this is pain on the y-axis. And the three graphs are three different alleles, so variants of one gene that is implicated with glucocorticoids response, so stress response, okay? FKBP5 is a gene that when it's activated, it allows the glucocorticoid receptor from the cytoplasm to go into the nuclei and start transcription, okay, so to influence the stress response. So what you can see by combining four different things, pain, hippocampal volume, the genotype of this gene, one variant or the other, and the adverse effects early in life, what they saw is when you have one allele, a protective allele, even if you have multiple trauma, you don't really see, and your hippocampal volume can change from more or less gray matter volume. If you have one protective allele, that doesn't change much in how much pain you experience. If you start to have one allele that is non-protective, you start to see a difference. And the more trauma you have, the more pain you have, especially if your hippocampal volume is reduced. And when you have both alleles that are favoring an altered stress response, then the effect of multiple trauma is even higher. Okay, so this is combining, it's an example of gene-environment interaction. Okay, so gene-environment interaction is one type of interaction that can happen in how we process information and how our biology works. There are other mechanisms that can mediate the interaction between the environment and gene expression, and these are so-called epigenetic mechanisms. Now, we don't have time to go into them, but at least I need to explain what DNA methylation is, because then it's important for the next couple of slides. And so DNA methylation, as you can see here, is DNA is trying to be transcribed, okay, by transcriptor factors. If everything goes okay, as it should be. Transcriptor factor bind to the DNA, they start transcription, and you see RNA that is produced by transcription. If you have DNA methylation, so if you have a methyl group that binds to specific sites of the DNA, this creates a physical barrier for transcription factors. They cannot really bind, and transcription is inhibited. Okay, this is a very simplistic, very basic understanding of how DNA methylation can influence transcription. And we know that some epigenetic modifications are stable, and they have to be stable. So you probably know that all the DNA in each cell in our body is exactly the same, but you can tell that a skin cell, it's very different from a neuron, from a muscle cell, from a liver cell, and so on, okay? So how come that from the same DNA we have such different cells with such different functions? It's because of epigenetic mechanisms and modifications, DNA methylation that are strategically distributed on the DNA, and so they silence a number of genes and let other genes be expressed. So, of course, in a muscle cell, you want genes for active and actin and myosin proteins to be expressed, but you don't want proteins that are normally expressed in neuron to be expressed. And the other way around, in neurons, you don't want actin and myosin, you want the neurofilament 200 and the peptides and so on, okay? So, and this modification determine whether one cell from a stem cell becomes a muscle cell or neuron, skin cell, and so on. And you want those modifications to be very stable throughout life. You don't want them to change. You don't want a muscle cell suddenly becoming a liver cell halfway in your life, right? And DNA methylation and epigenetic has started to attract interest in the scientific community because, for instance, we know that now DNA methylation changes is a hallmark of cancer, together with genomic instability, angiogenesis, evasion from growth suppression, the inflammatory microenvironment, and so on. Now also changes in DNA methylation, specific changes in DNA methylation, are considered a hallmark of cancer. Okay, a normal cell would have normally quite some DNA methylation throughout the genome, while a cancer cell will have way less DNA methylation throughout the genome that contributes to genomics instability, for instance. A normal cell will have low methylation, so it would allow expression of tumor suppressing genes, while cancer cells, they normally have those promoter regions methylated, and they inhibit genes that are normally tumor suppressing, okay? Those kind of things. And so DNA methylation has started to be studied in other fields, also the pain field recently. An important point of epigenetic differences, epigenetic modifications, is that differences arise during the lifetime. So if you compare twins that they are very young, you will see that different epigenetic mechanisms are very similar to each other, but when they grow up, when they are 50, and you look for the same marker, epigenetic marker, you see big differences between identical twins. And so thanks to these differences that arise during lifetime that are related to age and aging, they built what's called the epigenetic clock, okay? So they measured DNA methylation throughout the entire genome, and with simple sort of mathematical algorithm giving different weights to different sites of the DNA methylation throughout the genome, not necessarily in genes that we know what they do, it's really throughout the entire genome. They found a group of changes that can predict health, that can predict the lifespan and health span. And when they tested with other measures of biological aging, like telomere length or the proteins or transcriptomics, etc., it seems that the DNA methylation clocks are better in predicting health span, lifespan, and they can also predict other biological functions in the body. For instance, by looking at the DNA methylation, you can predict quite well the immune cell distribution, for instance, whether you have more T cells than neutrophils or CD8 positive T cells and so on. So very useful in some cases. And I wanted to discuss briefly about DNA methylation and epigenetics, because remember the example that we have about gene-environment interaction with the gene variant that can help explaining pain in people with adverse effects and early in life. Okay, another biological layer and substrate for that can explain the link between early life stress and a number of conditions into adulthood is DNA methylation. Okay, they know that when there has been physical or emotional abuse in humans, in animals, they've done it doing separating the offspring from the mothers. They see that DNA methylation in stress-related gene change, which also change how the stress system reacts when a stressful situation is faced, and this can mediate the link between early life stress and, for instance, depression or psychiatric conditions and so on. In pain, we are not there yet. We are studying it. But the idea is that the same early life stress and the mediation of stress related to genes related to the stress response can then predict whether somebody develop chronic pain into adulthood. This is also what I wanted to just mention, that also DNA methylation, and in this case is using the epigenetic clock, because what I didn't mention was that when they build the epigenetic clock as a measure of our biological age, what they can do is to use the epigenetic clock as a biomarker. So you use that as an outcome measure, as a biomarker, to see whether your intervention, for instance, can change the biological age. And this is what they did here. Okay, so they, instead of looking at specific mechanisms in stress genes, they try to check whether stress have an influence on the epigenetic clock used as a biomarker in itself. And it seems that that is the case, and it seems that that is the case also depending on whether that sex differences are implicated here as well, where I don't know if you can see here, but only in girls there is this is 17 years old, there's a significant association between stress and accelerated biological age. There are many studies about epigenetic age and biological age published in the last couple of years. This is probably the last one that has been published this year, and it's also a big cohort. It's about 2,500 people. They're all 50 or above 50, so it's an adult population, aged population. And what they checked multiple lifestyle factors and how they influence epigenetic aging. And they used BMI, socioeconomic status, smoking and alcohol consumption, comorbidities, physical activity, and aging. Okay, and they saw that the lifestyle factors can indeed influence and mediate the development of chronic pain in these patients. We also tried to study DNA methylation changes in pain conditions. We did it a couple of years ago, and we found a number of genes that can be implicated where epigenetic differences can be found in people with pain versus healthy controls. And other groups are trying to apply the epigenetic clock in a chronic pain population. This is an image from the first study in 2019, but the same group did additional studies and in even a bigger cohort of about 4,000 participants. And this is what they found. They found that epigenetic aging is associated with clinical and experimental pain in either older adults or people with chronic pain and impacts. Okay, so because it's different having chronic pain or having a chronic pain that has a clear impact on activities and induces disability, for instance. Okay. There are a number of potential applications of DNA methylation in chronic pain, and it can help, of course, to understand pathophysiology. Depends what genes are differentially methylated. You can go more into a stress idea, or more metabolomics, or more mechanisms related to the neuroimmune mechanisms, and so on and so forth. But what's interesting at this stage is also patient subgrouping, because there are a couple of studies published a few years ago where they found that both in fibromyalgia but also CRPS, when they used either DNA methylation or microRNA or even brain imaging, and they tested healthy controls and people with fibromyalgia or CRPS, what they found is actually 3 groups instead of 2. One of them is including only patients. So there might be, of course, the diagnosis that we have for fibromyalgia, for CRPS are clinical, but there might be that by studying biological mechanisms, we can find a subgroup of people with the more homogeneous changes in biology, and that we can probably assess them differently, treat them differently, because biological mechanisms are different from other people that might have the same diagnosis if you diagnose them only clinically. So, to conclude part 3 and then go towards the end, again, the field is its infancy. Many results are promising, many others have been non-replicated, so we need to be aware of the limitation and challenges of these biological approaches. And people with chronic pain might show widespread changes in DNA methylation that can suggest biological age acceleration, and by understanding more about biology and DNA methylation and gene-environment interaction, we can maybe understand better chronic pain, have a better diagnostic procedures, patient subgrouping, and so on and so forth. So basically the idea, when you put everything together, is this one, right? There's some genetic predisposition, which we saw at the beginning is important, but there are other mechanisms that can be influenced by lifestyle factors, environmental factors, that what they do is they change how the systemic responses. So they change how our biological systems respond to the challenges that we face. Let's think about the stress system and how we react to stress. It's a biological reaction. If your system works differently, you are probably predisposed towards a disease more or less. Of course, it's not only biology. The part today was about biology and the importance that biological mechanisms and omics, genetics, epigenetics, transcriptomics, proteomics, metabolomics might have in understanding chronic pain, but there are also the clinical and psychosocial aspects of chronic pain that are really important. This is a big study, again, using the UK Biobank that can be explored, that can be used to answer many interesting and relevant clinical questions. And this group of researchers, what they did was they found 6 items that are able to predict the development, the spreading of chronic pain 9 years after the assessment. And these 6 items are about sleep, are about neuroticism, mood, life stressors, and BMI in this case. So what I tried to do was try to put everything in practice, and everything that we discussed from genetic scores and that can be used, sex, BMI, inflammatory markers, age acceleration, and so on. We can probably, in the future, have a model of this that I just made up. It's not published. It's not it's not a diagnostic tool or anything. Let me see if I can. Yeah. And by modifying the polygenic risk score, the depression score, the sleep quality, the inflammation, and DNA and epigenetic age, you can estimate in the future whether you have more or less pain. And if you change this, also that should change. If you have a lot of changes in the genetics, these also change, and there's also some treatment insights that you can probably suggest. And with this, I think I said most of the things that I wanted to do. Yeah, this was just to conclude that we are in the science community, as in the society in general. If we look at the bigger picture, we are in a very exciting times, I would say, where bioinformatics, machine learning, artificial intelligence is coupled with data from physiological and biological processes, right? If you think about when you are in front of your screen, you download that new app, and you don't care about terms and conditions. You just click agree because you want the app, you want the power, and you don't care about what the conditions are. But perhaps the same app is tracking your eyes movements, your physiological changes, and by registering and recording where you look, how long you look, what you focus on, and the differences that you have in your heart rate variability, pupil dilation, eye movement tracking, etc. They know about you more than you might think, right? Same thing is for research in biology. Bioinformatics, artificial intelligence coupled with genetics data, epigenetics data, proteomics, and so on will give rise soon, and it's happening more and more at a very high pace. Will speed up the understanding of how our body works pretty soon. That's it. Did I stay within an hour? I don't know.